O'Hanlon Cohrt. I am the editor with CRISPR Medicine News, and today I am joined by a guest, Dan Worthley. Dan is a cancer researcher and gastroenterologist at the Colonoscopy Clinic in Queensland, Australia. And today we're going to be talking about colorectal cancer, more specifically the challenges around detecting colorectal cancer, and one of Dan's recent projects that explored CRISPR as a potential solution to that problem. So welcome, Dan. I'm really looking forward to this.
Karen, very grateful to be speaking to you. So thank you very much for inviting me.
Yeah, this is going to be great. So look, before we get into the details of your project, why don't we just start with a couple of numbers? Could you just tell us what way things are going with colorectal cancer? What kind of numbers are you seeing?
So colorectal cancer roughly is about the third or the fourth most commonly diagnosed cancer. But in most Western countries is the second commonest cause of cancer death. So, for instance, in my own country, in Australia, there are about a country of about 23 million. There are 15,000 cases diagnosed annually, about 5,000 deaths. In Ireland, Karen, there's about 3,000 cases a year and 1,000 deaths. In the US, so it's about 3 to 1. So in the US, there's about 50,000 cases, 150,000 cases and 50,000 deaths. So colorectal cancer, in fact, has been slowly reducing actually in annual incidence over the last several decades, not dramatically so, but still being the second commonest cause of death. But it has been slightly reducing in most age groups, except for the under 50 age group, where it's actually dramatically increased in incidence over the last several decades, and is now, albeit off a lower range of competitive cancers, but is the commonest cause of cancer death in young people between 25 and 34. Mm-hmm. It's common. It kills an enormous number of people around the world, but it's just incredibly preventable.
Yeah, I think with the numbers that you're telling us there, you're painting a picture of a cancer that is actually still very deadly. So such a large proportion of people that actually get it are still dying from it, and I guess part of the reason has got to do with when these cases are being picked up. So in general, what is the main approach to detecting and diagnosing colorectal cancer at present?
So that's a great question. So there are three ways of reducing colorectal cancer death in our communities. So either you reduce it from ever developing, and that's really hard. They're the very important sort of public health initiatives around reducing lifestyle factors, reducing sedentary behaviour, reducing obesity, stopping folks drinking, reducing processed meat. All of those things are very difficult but very important, and are being tackled at a policy level. You can try and improve survival in folks that are diagnosed late, and a lot of my colleagues, and including in our lab, we've been working on trying to improve therapies in cases that are detected late, so you can improve survival in those folks. But the standard way that most places around the world have tried to tackle this is through screening programs that try and bring diagnosis left from later stage disease to early stage disease, or even pre-invasive disease. Yeah. And there are a whole range of initiatives that have tried to do that, and that's actually been pretty effective. It's been proven in randomised controlled trials that screening for colorectal cancer improves cancer-specific mortality, and that's why most developed countries around the world have at least a bowel cancer screening policy, if not a sort of a funded program.
Yeah. So when it comes to the actual detection, the major diagnostic procedure is the colonoscopy.
All roads lead to colonoscopy for diagnosis, but there are a range of different approaches. So the US promotes the US Multi-Society Task Force on Colorectal Cancer Screening, presents a couple of options. So they talk about either direct primary colonoscopy from folks who are 45 and older. So that's if you're an American, you get to 45, then one recommendation would be to have a colonoscopy, even if you didn't have symptoms or a family history.
Okay.
However, a lot of other countries, including Australia, have adopted what's called a two-step program. And so that's where they do a cheaper, more easily population-based delivered first step. In Australia, it's something called an 𝐓𝐢𝐦𝐞𝐫𝐞 fecal occult blood test. It's looking for microscopic bleeding in the poo, which often precedes symptomatic bleeding. And folks who are then positive go on to colonoscopy. And there are a range of those sorts of first step. Cologuard, a famous sort of stool DNA test, is also commonly being used, perhaps mostly in the US. But I think the 𝐓𝐢𝐦𝐞 test is perhaps the most widespread first step. So either direct to colonoscopy for a lot of folks, but in Australia, UK, New Zealand, and even as an option in the US as well, would be to have a fecal occult blood test. Does your poo have microscopic blood in it? If so, proceed to colonoscopy.
Yeah, that's the same procedure that's happening here in Ireland also. Yeah, but
and I think that's a pretty good. You know what? Each of them have their benefits and and costs, but you know most screening programs are plagued by a combination of convenience, cost, compliance being a huge one, and a really major focus for how we can improve, and then complications. And so when you sort of try and balance all of those things, you know what, high effective colonoscopy can be delivered well, but for a lot of folks, they think that the balance lies in that two-step sort of program.
yeah. So you mentioned there that you're moving the detection, I guess the stage of disease to the left by implementing screening programs. So how early in general are you now detecting colorectal cancer? And you mentioned sort of some of the pros of the screening programs, but are there also limitations to the current two-step, we'll say the two-step program that you just described?
So in terms of the detection, you know, the goal of detection, yes, is to bring it as early as possible, or even in what are called advanced pre-invasive lesions, so big important polyps that can be cured at colonoscopy. The two-step programs with fecal immunochemical testing, for instance, their sensitivity on a one-off test is about 62 or thereabouts, 60% for bowel cancer,
Okay.
and slightly lower for advanced pre-invasive lesions, about 24% or thereabouts. But often these programs are delivered serially in Australia every 2 years, most places probably every 2 years or annually. And because bowel cancer progresses at such a relatively slow rate, that you sometimes get a couple of bites at the cherry in a screening program, particularly through advanced pre-invasive lesions and into early cancers, so that if you don't find it the first time, then maybe you'll precipitate it in the second time without losing too much in terms of the stage of diagnosis. But having said that, in Australia, where I'm most familiar with stages, but I'm sure it's reflected in Ireland and the US and Western Europe, is that about, you know what, it's about 75% are diagnosed in the loco-regional stages. So these are stage 1, superficial part of the bowel wall; stage 2, still in the bowel wall but a little bit deeper; stage 3 then is lymph nodes, requires chemotherapy after surgery, but these are still not early stage disease, but relatively early stage disease. And then stage 4 which is when it's spread to the liver or the lungs, accounts for about 25%. So if we could, so it's about 25% per stage. So if we can all of a sudden reduce that 25% down to 10%, down to 5% by shifting it left, which is the whole premise of screening, then all of a sudden we have a much better chance of curing folks, either just with surgery alone for 1 and 2, or with chemotherapy for 3.
So I've been reading that part of your goal is to ensure that no Australian dies of colorectal cancer. That is your ultimate goal.
Do you know what? I think it was. I was challenged, and when I came back to Australia, I had, in fact, it was an infectious disease physician who came to me and said, "Can bowel cancer be eradicated completely?" And it was actually quite a provocative question. This gentleman, in fact, his name was Steve Wessling. He'd been involved with a whole lot of HIV work in, Africa and in other places where they had these types of ambitious goals. And I sort of initially thought, well, do you know what? That's infectious disease. I'm working on a non-communicable disease. That's sort of like heart disease and other cancers, albeit although sort of cervical cancer should be eradicated soon. But but what that challenge gave was, then you thought, well, okay, well, is it eradicable? And how might we map out a path there? So eradicating bowel cancer death is really hard. However, we went through and with a whole range of different specialists mapped out what it would take in order to reach that goal. Not to show that it was easy, but to show that it existed. The destination existed. So, and once you have that type of approach, all of a sudden you're thinking about each of these other stages and approaches is altered. And so I think it's helpful to have as an aspirational goal, understanding that it's going to be incredibly hard and require new tools and new approaches to get us there.
Yeah. So on the topic of new tools, so you were recently involved in a project that actually I won't say attempted, it did it actually. You developed a new tool to detect cell-free DNA from colorectal tumors in the guts of mice using CRISPR as part of that tool. Perhaps you could tell us about this work.
Well, thank you very much. Yeah. So, and I gave, you very kindly invited a talk on this topic late last year. This project was a great collaborative project between my team, along with Susie Woods and Josephine Wright in Australia, and Jeff Hasty and Rob Cooper primarily, albeit with others from UC San Diego. So Jeff Hasty, who's a synthetic biologist, is just a superb scientist who has an incredible lab at UC San Diego. I was visiting his lab meeting, and he very kindly sort of does a run through when you visit his lab of his students' projects. And one of them was Rob Cooper, who was working on horizontal gene transfer. So this is the concept, and I know for this audience, not too important to break it down, but usually there's vertical transmission when one cell divides to give 2, you replicate DNA. But of course, there's also horizontal gene transfer, particularly common in in bacterial life, where genes can be passed for fitness or for nutrition horizontally from non-parental offspring relationship. And Rob was very interested in this in antibiotic resistance, and he showed this great movie. The Hasty Lab always shows this beautiful self-evident science where you can all of a sudden, within a frame or two, both with rigor but also just understand the whole premise of the experiment. And they showed it was 2 bacterial species. It was Salmonella and Acinetobacter bailey, which is a renowned competent bacteria, naturally competent bacteria that hoovers up DNA, and showed how it would steal genes from the green fluorescent bacteria in the well next to it. And I saw this, and we're all trying to pitch Jeff ideas about things that he could work with us on. But one of the obvious things that it looked very much like, it was 2 cells in a dish. They happened to be both bacterial, but why? And one was taking DNA from the other. But perhaps we could do this with cancer cells, and a bacteria was the sort of the premise. And so we spoke about that after the lab meeting. And we developed a bit of a plan about how we could improve efficiency using, long homology arms, about how we might be able to facilitate natural competence of Acinetobacter bailii, how we can actually probably do some of the earlier steps before natural detection just to really test the proof of concept, including some donor DNA that we were able to engineer into some of the cancer cells for the preliminary experiments. And we happened to be using a mouse colorectal cancer model that seemed to be sort of custom made for testing this. So that was the sort of the genesis of it. We then, you do ambitious projects slowly with sort of go, no go type steps. So we initially engineered a Acinetobacter bailii, so a famously naturally competent, highly competent cell, bacterial cell, with long homology arms within its genome, and linked in between those homology arms. The homology arms were for an important oncogene called KRAS, that's present in about 20% of colorectal cancers. And then in between it, we had both a green fluorescent protein and a KRAS, a kanamycin resistance gene that had 2 stop codons within 8 base pairs of it. So the Acinetobacter bailii, I'm sorry, this is getting into the weeds a little bit here.
Oh, okay. That's what we're here for.
The Acinetobacter bailii didn't express green fluorescent protein and wasn't resistant to kanamycin because of these stop codons. However, if it received a donor template of DNA from a cancer cell, it would be able to repair that kanamycin resistance gene and all of a sudden be able to be selected for and would become green. Yeah. So those initial experiments were done with sort of plasmid DNA initially, and then genomic DNA, and then cell lysate, and then co-culture. All of those were using a donor DNA within the cell line, the cancer cell lines. And then we accelerated that to the mouse model, where we injected via mouse colonoscopy cancer cells into the mouse colon, and via delivering the Acinetobacter bailii biodetector cells, we were able to detect or discriminate mice with and without colorectal cancer. And then the only final step, I'm sorry, is that that was very good, but those experiments that we published required donor DNA from the actual target cells, which is not helpful in terms of a clinical application. And then so we slightly rejigged it. We then had in the bacteria 2 homology arms with a repressing gene in the middle of those. That would be repressing a selection cassette elsewhere in the bacterial chromosome, and then following horizontal gene transfer and homologous recombination, it then kicked out the repressing gene that then allowed the repressed selection cassette to be able to be selected. Then that's where the CRISPR bit comes in. Yeah. So this is that. All the rest of that's commentary. CRISPR is obviously the star of the show. So that's when Rob had this very cool idea, and so it was entirely Rob's idea, this part, and it really was the only way that we could make it work, is that we really wanted to discriminate between mutant KRAS and wild-type KRAS, because it wouldn't really be valuable to detect wild-type KRAS. And so what Rob had been working on, endogenous, it's a type 1F CRISPR-Cas system that Acinetobacter bailey uses. And what he said was, well, I'm using this in other applications. What we could just do is define if the G12D mutation overlines a PAM site, what we can do is then mean that the mutant DNA would obscure the PAM site, and therefore we wouldn't actually cut at that point. And so when a mutant one came in, we couldn't cut. When a wild type KRAS came in, it was cut, and that would then kill the cell. It would cleave the DNA and actually kill the bacteria, so they wouldn't. And that, in a very large nutshell, I'm sorry, was the paper.
No, I think you've done an amazing job of actually taking us through more or less the full study without any slides. But just to help our listeners, we will put up in the comments section of this, we will actually put up a link to your publication, which was published in Science, and also a link to, we interviewed you last year also on this story, and also a link to the webinar. So people have both the original publication, the interview with some figures, and also the webinar where you very elegantly showed us with your slides this whole process. As you say, there was a lot of steps involved in the process, but you reached the point where you were able to show that your system could discriminate between mice who had the tumor and didn't have the tumor. So it's working.
Yes.
So obviously this is something that you're going to continue progressing with. I'm conscious of our time as well. I want to ask a couple of questions about how this could be translated to humans, even though I know, many steps have to be done before that. So perhaps you just tell us where you are now.
So it's a very good point. So what we've been working on a little bit to have a workable clinical prototype is that we've so KRAS G12D is one mutation. It's a really important mutation, but we were only detecting one mutation, and that won't really cut it in terms of being utilised for colorectal cancer detection. So the next step that we've really been focusing on is multiplexing. So trying to piece together in some iterations in a logic gate type way. So if KRAS, then this, if KRAS and additional mutations, then that. We're getting up to 2 and we're trying for 3. Probably we really would be liking to have a 4, at least a quad, a 4 multiplexed system. We are, with this technique as well, we are limited to hotspot mutations, at least the way that we've currently envisaged it. So there are a whole range of different important colorectal cancer events that occur, but some of them are loss of heterozygosity, or that wouldn't, or at least the way that we currently understand it, we wouldn't be able to utilise that. So, but that's been the next step that we've done, and we think that even outside of the cancer space, to have a multiplexed bacterial biodetector for different forms of DNA would be very valuable, even in infection and other things. So we think that that's an important thing to invest in, and that's what we're working on at the moment.
Okay, great. I mean, absolutely. I mean, so the more mutations you can put inside your assay, the greater percentage, I suppose, of cases that you may be able to detect.
Precisely.
That makes a lot of sense to me. So you're using a totally non-pathogenic and very well characterized species of bacteria, but I'm sitting wondering if it would be possible to translate this to the existing microbiome that we already have.
That's a really awesome question. So, what are the the already colorectal colonizing competent candidates. And that's one of the other co-authors is an incredible gentleman called Rob Knight, who is a microbial ecologist and computational microbiologist at UC San Diego. And with him, we have been looking for new competent candidates within the microbiome. They would then need to be engineered. We don't have a lead candidate at this time. That's the but we're looking at that in parallel, because Acinetobacter bailey, do you know what? It was used because it was well characterised and it was competent, but it's a soil organism. It is not necessarily native to our bowels, and will probably be difficult if we wanted it to colonise in order to achieve it. Perhaps we don't. Perhaps that would be its exoticness it's one of its advantages. But I agree with you that's one sort of key avenue of that we're looking at, is to try and find, well, what are the ones that actually already exist and are competent anyway. So great question.
Okay. And how would you, so in your mouse study, the bacteria was administered rectally?
Yes.
Obviously, this is not going to be the route of administration in humans. So how would you envision doing it? A pill or something, a capsule?
The only thing that's, there are a whole range of things, so I don't want to dismiss other people's work in other different scenarios, but I think the only way that you deliver probiotics, at least for gastrointestinal disease. So I think for cancers, there is still a whole range of opportunities for intratumoral delivery via CT guided stuff. But if you want to address the gastrointestinal tract, it has to be done orally.
Yeah.
So we haven't solved for that yet. So it'll be a range of possibly new candidates, but also packaging, I think, will be the key aspect. There's a whole range of enteric type medication coatings that are used. We haven't solved for it, but we identified it as incredibly important.
Yeah, because you, of course, want people to comply and want people to want to
exactly.
take it. I mean, nobody's going to take part in a...
There's one catch. Doesn't really work. That's right. It has to be delivered rectally. No. So that won't...
Forget it.
Yeah. So, and all of a sudden, the benefits over colonoscopy don't really seem to be too profound. So
no, exactly.
We agree.
Yeah. So I also read in some of your work that it could also be an idea to detect directly in the stool, adding an engineered bacteria to the stool. This would obviously solve a lot of the problems in making somebody ingest an engineered bacteria. It would probably solve a lot of the regulatory issues as well.
Yes.
How would you bypass the issue of the degradation of cell-free DNA that occurs before it gets to the stool?
Yeah, it's an excellent question, and it's one of the advantages of delivering it endogenously, is the fact that there is stool DNA degradation, and it does interfere with detection. Stool DNA is a widely, relatively widely employed approach now already, and so I mentioned Cologuard before. In the New England, just yesterday, there's this Blue Sea study, which by Imperiali and al. That is a new sort of next generation multi-target stool DNA test. So there certainly is tumour DNA within the cell, within the stool.
Okay.
I agree, though. As soon as if you are using a living cell, then the advantages over other ex vivo tests are lesser than if you're delivering it in order to detect and react endogenously, which is a living biodetector has real value in the fact that it can detect, process, and respond. That's where it really has a level of difference from other ex vivo tests. Ex vivo testing, the only other way it can be competitive against some of those things, so a living cell, is in resource poor places, I think, where you are removed from laboratories and other things. But do you know what? Detector and Sherlock and some of these other relatively straightforward CRISPR approaches that are ex vivo, may have us on toast there, as really have probably a significant advantage over a living cell anyway. So, I think as an ex vivo test, we, at least at the moment, are not enormously competitive outside of very resource poor circumstances or geographically remote circumstances. One of the topics that we haven't really isn't mature, but we've explored is in rural and remote Aboriginal Indigenous populations in Australia in terms of Group A strep. There could be opportunities there where a living cell is able to deliver a signal to a swab on site to guide antibiotic therapy. We are not there, Karen, but these are some of the lovely discussions that you have when you're sort of trying to dream up the next project.
Yeah, absolutely. Look, I think we're sort of running out of time, but you are co-organising the Microbial Healthcare Symposium, which will be coming up in Brisbane this July. And as I understand it, there will be many very experienced scientists, medical doctors also there to discuss the many ways in which microbes can be used to help in the detection, the treatment, or in any other way improving the situation around cancer. So
that's very kind of you, Karen. So
go, but I don't think I can make it over. I still have that on the list.
So microbialsymposium.com is the website. So it's the 11th and 12th of July. We have Jeff Hastie, who is the senior author on the paper, Rob Knight, a co-author, two stellar scientists, and Siddhartha Mukherjee, member of the National Academy of Medicine, Pulitzer Prize-winning author and oncologist. And he was one of the guys that really established this concept of life healing life, so cells being used as for detection and for therapy. And I'm sure we're in an exciting field, not just in bacteria, but especially in bacteria, but also in other living cell therapy. So it'll be a great meeting. Thank you for letting me.
Thank you so much for your time, Dan. I think it'd be great to stay in touch, and I'd like to follow this project as it progresses. And perhaps when you get to a later stage, you'll come back and give us an update.
I'd love to do that, Karen. Very grateful for the invitation, and thank you for your audience for listening. Thank you.
Thanks to everybody. And I'll put up a few links of the things that we discussed today. And I wish you the best of luck with all of these really exciting projects going forward.
Thank you so much, Karen. Have a good day.